Alright, I think this is where we got to. Started discussing ATP, energy molecule. So, when we're talking about metabolism, we've got two options. It can be aerobic, okay? That means that at some point in the process I have to have oxygen available for the process to continue, okay? Or it can be anaerobic, which means I don't need oxygen available within the cell for metabolism and ATP production to keep going. Right? So, how long then I'm moving for, how long I need energy for, is one of the prime factors that decides which energy system I'm going to use to make the ATP. If I'm doing something that is very quick, very powerful, then I'm going to predominantly use systems that are anaerobic in nature. All right? So that could be weightlifting, that could be throwing, jumping, short sprints, would be anaerobic. And we've got two systems that work anaerobically. If I'm going to keep going, so if I'm going to keep moving, or, interestingly, when you're sitting here in the classroom, you're working aerobically. You're just working at a very, very low intensity, but you sit for a very long time, right? So you have to make energy somehow while you're sitting there. So because it's 45 minutes of listening to me wittering on, right? That's an aerobic form of making energy to keep you warm, to keep your digestion going, to keep your heart going, to keep you upright in your chair. All of that is aerobic. It's just very low intensity, right? So when we look at where the switch in the predominant system occurs, somewhere around 3 minutes. 2.5. Used to be 2.5, but most of the textbooks now are saying 3. I think that's important. We're learning, as I said, at the beginning of the semester, XPhys is probably the area of kinesiology, sports science, that is changing the fastest. All right, we're learning so much more about how the body works. We've got better measurement techniques. We've got better access to clever equipment. And so what we know about how things work is changing all the time. It also makes it difficult to pinpoint on some things exactly what's going on all the time. So part of that learning curve is the fact that we don't know everything yet, and we get some mixed results sometimes. Okay? Then click up. I should tie it to my hand. So, system number 1 then, if I want to change intensity, right? So if you want to get up and walk out the room, right now you're ticking over, it's very low, okay? And you're not using very much ATP at all. If I want to stand up and leave, then I need a suddenly, dramatically larger amount of ATP in order to stand up and walk out. Right? I can't stand up at the intensity I'm working at now. So when that happens, how do we do that? What's that first immediate way that we can make a bit more ATP when we need it? Right? So initially, we've got some ATP just sitting in the muscle cell. Okay. So I need a lot of energy very quickly. I've got ATP right there that I can break down, release the energy bond for muscle contraction. Right? But it's only around 5 to 10 seconds worth, depending upon the individual. So 5 to 10 seconds doesn't get me very far, really. So ATP that's available to break down straight away when I need it is for a very, very brief amount of time. So, okay, what happens after 5 to 10 seconds? Well, I haven't had enough time yet to be breaking down a food molecule. Even though we said carbohydrate was a relatively small molecule, it still takes time to access the carbohydrate and start to break it down. So 5 to 10 seconds isn't enough time, so I still can't get to food to produce energy. Right? So the next part of system 1 is another energy molecule that just sits in the muscle cell called phosphocreatine. And it's an indirect but very quick, immediate source of energy that I can access. Okay? Because I can break down phosphocreatine, kind of borrow the energy bond there, rebuild the ATP, and then break down the ATP and have energy available for contraction. It would be very useful if we could take the energy directly from phosphocreatine, but that's not how humans work. Right? So phosphocreatine within the muscle cell, somewhere around 15 to 20 seconds worth. Right? So if I add those two together, the immediate energy that's available in the cell is somewhere up to around 30 seconds. Okay. So I have ATP, right? And that's going to provide me with energy. So I'm going to break off an inorganic phosphate, release that energy bond. So that leaves me with ADP plus the inorganic phosphate I took off. It releases a hydrogen, which is problematical because hydrogen's acidic. And I have energy release that I can use for muscle contraction. For 5 seconds. So part 2 is I have phosphocreatine, which has an indirect source of energy I can get at. So the first thing I have to do is get at that energy. Right? So I'm going to break down phosphocreatine into creatine plus an inorganic phosphate plus energy. Right? And then I can use that energy and that inorganic phosphate to rebuild my ATP. Does that make sense? Eventually then, there would be a risk that I would finish up all my phosphocreatine as well. So we also have to have a mechanism to rebuild the phosphocreatine. And later in the semester, we're going to spend some time talking about EPOC. Okay? In order to rebuild the phosphocreatine, I have to use aerobic metabolism. Right? So that's why we need a recovery phase when we've done a really hard sprint. I can't sprint for a very, very long time because ultimately I don't have enough energy source for that. Right? So then what we can do is borrow this, let's do it with another color. I can make ATP aerobic way, break it down, borrow that energy, and rebuild phosphocreatine. In order to do that then, I have to either decrease the intensity that I'm working at, so when we do interval works, we sprint and then we walk, and then we sprint and then we walk, or we jog, right? I have to have a period where I'm not sprinting. Or this is a recovery phase, right? And we have a couple of enzymes that help us out with this system. The first one is ATPase. Easy, right? The A's tells us it's an enzyme. The front part of the name of the enzyme often tells us what it does. In this case, it facilitates breaking that inorganic phosphate off ATP and releasing energy. Remember, enzymes don't cause the breakdown, they facilitate breakdown. And here we have creatine kinase. So, system one, the first port of call, if I need a quick, immediate source of energy, so it could be a sprint, or a throw, or a high jump, or getting up and walking out to the car, right? Anywhere where I'm going to suddenly increase intensity, ATP-PC system is going to kick in and provide that ATP for a short period of time to kind of tide you over. Okay. If you're a 100-meter sprinter and you're good at 100 meters, ATP-PC system is going to be your predominant energy system for your entire event, right? Because if I'm good at it, I'm going to finish 100 meters in under 30 seconds. Or about 200 meters. If I'm really good at 200 meters, that's going to be predominantly ATP-PC. Not for most, not for me, not for most people, but if I'm really good at it. Okay? Questions on this idea? All right. So then it would be somewhat logical to say, oh, okay, then when I'm sprinting, when I'm running as hard as I can, or I'm lifting a very heavy weight, the reason I get tired, the reason I get fatigued, I get to a point where power, speed, strength start to drop down, right, would be because I ran out of ATP or PC in the muscle cell. Okay. That's logical. The research doesn't tend to support that argument because the timelines are different. When you look at how long it takes to run out of ATP and how long it takes to run out of phosphocreatine, generally fatigue happens a little bit later than that. So this doesn't seem to be the main cause of fatigue. Okay. If I'm going max effort all out, which really is not a realistic event, right? No one goes max, but you can deplete ATP-PC in about 4 seconds. Okay. So I suspect they test that by chopping up a cat muscle and innovating it and seeing how long it takes until there's nothing left. I suspect they don't find that out from a human. Right? So that's not the main cause. Okay? The main cause then seems to be this bad guy here, our hydrogen. Because when I'm breaking down the ATP, I'm releasing hydrogen cells. Hydrogen is acidic. And we said our enzymes don't like it if I increase acidity. And so because I'm releasing the hydrogen, ATPase and creatine kinase start to slow down. And then ATP production starts to slow down, and we see a drop in power, strength, speed. Measures. All right. Another factor is this inorganic phosphate that gets released. That there's some indication in the research now that the inorganic phosphate somehow interferes over time. And there's also some data that says, well, where was the ATP, like the muscle cell? If you think of the muscle cell as this room, where in the room was my ATP when I needed it? Right? If it's all over here, and the thing I need to use the ATP is all the way over by the door, then that might cause a problem. Right? So as I say, the finding, this stuff is changing all the time. I can guarantee you guys that in 3 or 4 years' time, what we know about what causes fatigue in power events, there'll be tons more information. So you need to stay, if that's where your career is going to be, you want to stay on top of that because it'll change how you approach a training program. So, we already mentioned we need an aerobic recovery period to rebuild because I need energy now to put all the PC back together for the next time I need it. And this is also where people think about, well, maybe I should take extra creatine then. If I had more creatine, phosphocreatine in the cell, then I would have longer to access the energy bond and make more ATP. And so that's why people look at possibly taking a creatine supplementation. And we'll come back, I think it's chapter 15 in this edition of the book, is about ergogenic aids, right? What are things we take and do that benefit performance, or that we think are going to benefit performance, okay? So ergogenic aids could be steroids, but it also can be creatine supplementation or caffeine. Okay. So, but there's a little piece of information on page 34, and there's some caution to be used when you're looking at creatine. Right? It causes water retention, and so in the short term, you see an increase in weight. Could be as much as 6 pounds, right? So it doesn't take very many days of taking creatine for you to put on some water weight. Well, okay, it's water weight, not fat weight, but it's still weight and you still have to move it. So it can impact performance the wrong way, right? It could slow you down a little bit. instead of making you able to go for a little longer. There's also, doesn't look as though very much of the supplement actually makes it into the muscle cell. And once it's made it into the muscle cell, right, it still has to be turned into phosphocreatine to be any use within the system. So there are some question marks when you look at the literature about creatine supplements on how beneficial they may or may not be. There is, over time, some improvement in performance, but it's generally very, very small. Very small, like less than 10% improvement in performance. So you've got to look at who would bother to take this. If I'm going for world records, that less than 10% might make a difference. Right? If I'm trying to win the high school state meet, that 10% probably isn't going to make that much difference. So who would I encourage? If I'm coaching, okay, do I suggest this or do I just not bother mentioning it? And I think it would depend who you're working with. I read a paper last year that gave some pretty strong evidence that creatine supplementation in older women helps maintain or increase muscle mass. Well, that's pretty important as you get, right? When I'm 60, I want as much muscle mass as possible because I don't want to fall over. So that might be quite interesting to me. So I think you've got to use a little bit of caution. Also, if there's any history of any kind of renal problems or kidney disease, you should not be taking creatine because it stresses the kidneys. So if we're looking at this picture, if I had more substrate or more enzymes, or enzymes that were working where the action was faster, I might be able to make more ATP. That would be the beneficial picture I would hope to see by training. Right? Particularly if I'm like a basketball player. Okay? Basketball, short, sharp bouts of high intensity, and then kind of standing around or jogging around much lower intensity. So basketball on the whole is probably more anaerobic than aerobic, even though it goes for quite a long time. You don't go hard all the time, whatever coach asks for, right? That's not the reality of a basketball game. The whistle blows, we stop, we all jog back to position, the ball starts, we run really fast. I may or may not be defending someone, so I'm kind of hanging around waiting to see what's happening. Right? So predominantly aerobic, stop-start, repeated bouts of high intensity, going to use this system quite a lot. So if I'm training, adaptation comes with training. It's why we train. If nothing was going to change, there would be no point in training. Right? The whole point of it is that I expect to see improvement, and a lot of the improvement is change in biomechanics, change in understanding of the skill, change in the motor control and coordination of the skill, and a lot of the improvement comes from physiological adaptation if I do the right kind of training. We can get it really wrong and we won't see improvement in performance at all. We may even see a decrease in performance. If we get it right, then the adaptations that we would be driving, that we would create, should benefit. So if I'm looking at my enzymes, right, then it makes sense that if I've either got more of those enzymes or the enzymes are more active, that would help me produce energy. Okay. The literature, when we look at adaptations, is really irritating to me. It's very equivocal. Depends which study you're reading, it depends how they organize the study, how did they define sprinting? Was sprinting a one-off sprint, or was it a sprint and a rest and another sprint? How are they measuring in the study, right? So there's lots of different variables that alter the outcomes in these studies. So right now, the picture's a little hazy. Particularly in creatine kinase, quite a bit of the literature suggests that we see creatine kinase increase in activity, which would allow me to make more ATP. So that would be very beneficial. Adaptation is typically positive. Not always, but typically. Adaptation is going to be something that is useful, right? The body's way of changing so that the training is no longer stressful, and then you have to up the stress level. Okay. There's some information that suggests we see faster regeneration of ATP, which would obviously be very beneficial. And so those two things would theoretically increase performance in short duration, high intensity activities. What about stores of ATP and phosphocreatine? So again, if we could increase how much was already in the muscle, then I would be able to make more ATP before I fatigued, in theory. All right? So we do see some increase. Tends to be longer periods of study. So if I just do a 6-week training session, which is a 6-week training program for the study, we don't see the concentrations go up. But if I do a 6-month study, we do see concentrations go up. So it takes some time, it appears, to impact the actual storage of these molecules. Endurance training has no effect. Well, that shouldn't be surprising because we don't use this system for endurance training. And so why would this system adapt, right? If I do bicep curls, I don't expect my quads to get stronger. That would be silly, right? So if I'm not doing exercise that uses this system, I wouldn't see change in this system, okay? And then just to complicate the picture, there are studies that look at improvement in performance and where they don't see the change in the concentration, but they do see improvement in performance. So then what caused the improvement? All right. Depletion generally doesn't occur. So if I never deplete this in a sprint, why would I need more of it? Right? So, as I say, it's a little hazy right now because we're getting the opportunity to look at things a lot more closely. It's all very well looking at a frog, right? How many of you chopped up frog legs at school? Oh my God, you did cry. You did, yeah. That was like my favorite lesson. I felt bad for the frog, but it's so cool. You pull the leg out and you give it a little bit of electricity and the muscle goes, and the leg isn't attached to the frog anymore, and you can make the muscle contract. It's like, oh, that was one of my favorite experiments in school. Alright? So, it's all very well looking at a frog, but we're not frogs. Okay? So right now it's becoming very interesting because we're starting to be able to actually look at people. I can't chop someone's leg off and innovate it in a lab. So how do I know what's going on in a person versus a frog? So until we get the ability to look at people while they're moving, this stuff is going to stay really fuzzy. Okay? But this is what we know right now. Obviously, we've got quite a lot to learn, I would say, since we've got some studies say yes, some studies say no. Seems to depend on what the activity is. Okay. Questions on system number 1? Short, up to 30 seconds. Okay. I've got to have a way, if I want to change intensity, I've got to be able to suddenly create ATP faster than I have the ability to break down carbohydrate. And that's the role of system 1. Right? ATP-PC system is an immediate source of energy right there in the cell. Okay. So, I get to 2.5 minutes, 3 minutes. Sorry, I get to 30 seconds. I want to keep going. What do I do? How do I make ATP? How do I produce energy for muscle contraction once I get to that 30-second mark? Okay. So we have to transition into system number 2, glycolysis, and it's not that system number 1 necessarily turns off, but system number 2 now becomes predominant because I've had enough time to start accessing blood sugar or stored glycogen. Okay. So I can start breaking down molecules of carbohydrate and getting at the energy bonds within those molecules and making ATP. So, in your book is this nice ugly figure over here, okay? And this is where my teaching evals go through the roof, right? You don't have to learn this, okay? We have to know some things about this. There are a couple of things that I'm going to highlight that are really crucial to your understanding of what happens in adaptation and when I'm training and trying to use this system. But you don't have to learn every single, it's a 10-step process, right? Every step, we said enzymes are very specific, they only do one job. That means at every step I have a new enzyme, and I get a different molecule, and I keep breaking it down until I get to the bottom, right? We don't need to learn that, because I've done that many times in my career, and I don't know it, so it doesn't work. So let's not waste time on it, okay? That's why we have textbooks and the internet, and we can look it up, okay? We have to know what we're looking for, though, so you've got to understand some of it, right? ATP-PC system and glycolysis both take part in the sarcoplasm of the cell, so in the jelly bit of the cell, okay, in the watery part within the muscle cell. And right now we are going to say, glycolysis is anaerobic, period. It does not need oxygen available in the muscle cell for glycolysis to occur or to make ATP. So often when you look at the internet or if you look at a different textbook, they will term glycolysis as anaerobic glycolysis or aerobic glycolysis. And I understand why they do that, but to me that is really confusing. Because aerobic glycolysis suggests that I need oxygen here, and you don't, ever. The aerobic portion is what happens to the carbohydrate after glycolysis. Okay. So glycolysis is anaerobic. I do not need oxygen available, but what happens at the end of glycolysis will depend on whether or not there is oxygen. So, here we go. Glucose, glycogen, both our carbohydrates. Right? I've got glucose in the bloodstream, I can shift into the muscle. I have glycogen sitting in the muscle that I can start to break down. Okay? And we'll come back to this idea in a minute. All right, tell me if you can't see this because we might leave the lights on. Okay, so here we go. Here's glycolysis. This is the version we want, right? Not the complicated version. My version's much nicer. Right? I need carbohydrate. So that is going to be glucose or glycogen. How many carbons are in glucose or glycogen? Six. Okay. 6. So carbohydrate is a 6 carbon, a simple carbohydrate is a 6 carbon molecule. Glucose and glycogen, or glucose is a 6 carbon molecule. All right. When I feed glycogen in here, it actually feeds in as glucose 6-phosphate. Glucose, it's not exactly glucose, but, all right. So what is going on here is working with a 6 carbon molecule. Okay. And at each step here, we're going to break it down, break it down, break it down. When we get to around, I think that's step 5, we split the 6 carbon molecule into 2 3 carbon molecules. Yes? With me so far? Okay. So we break it down, we break it down, we break it down, we break it down. We end up with a 3 carbon molecule called pyruvate. But we end up with 2 pyruvates because it's 3 carbons out of 6 carbons. Okay? Make sense? Everybody okay? I know it's Friday, guys, but I need to know. Good? All right. So, well, that's great, but where's the ATP part of it? Right? So, where's the actual ATP in all this?
Right there.
Okay. So when we are breaking down our 3 carbon molecules here, we make ATP at a couple of different points. All right, if you're looking at the complicated version, it's at 2 different steps. Don't worry about it, right? I make 2 ATPs for each 3 carbon molecule. Right? So I make 2 plus 2, 4 ATP for every 1 6 carbon molecule. I don't end up with 4. 4's what I make, but in glycolysis, I actually have to use some ATP in order to get it going. Right? So I lose an ATP here before I break it into 2 molecules. glucose in from the bloodstream, rather than breaking down glycogen, I also lose an ATP during that process. Can you see this on the camera?
Yep.
Hence, this. Right? If I use blood glucose, I lose an ATP, I lose another ATP, I make 4 ATP, I end up with 2 ATP. If I break down stored glycogen in the muscle cell, I only use 1 ATP, I make 4, my net effect is 3. Right? So, this is a little bit quicker to feed in, because this has to go through some jiggery-tokery to turn into a glucose. But from an energy standpoint, this is more useful because I'll get more ATP from it. Right? So stored muscle glycogen is important. Now, one of the things that makes glycolysis a problem if I'm an anaerobic athlete is that we release some hydrogens during glycolysis. Okay. The hydrogen itself doesn't directly cause much of a problem, depending on how quickly I'm making the ATP. The hydrogen gets picked up by a molecule called NAD, which is a bus. Okay. So NAD acts like a bus. It collects the hydrogen, and then what it does with the hydrogen kind of depends on whether the oxygen is available or not. If it can, it's going to take that hydrogen somewhere else in the muscle cell, and we're going to use it to make ATP with oxygen. Right? So that's chapter 3. We'll come back to that idea. If, however, there's not enough oxygen in the cell, then the NAD bus is going to pick up the hydrogen and it's going to shuffle it around, and it's going to get picked up by pyruvate. Okay. Pyruvate turns into the dreaded lactic acid. Okay. So it takes the hydrogen, turns into lactic acid, lactic acid doesn't remain lactic acid. It's hugely unstable. It basically doesn't remain lactic acid for any time whatsoever. It turns immediately into lactate, is shuttled into the bloodstream, moved off around the body, turned into glucose via a process that we call the Cori cycle, which I typically don't do with undergraduates. So you don't have to worry too much. But it's a really interesting idea, because when I was in undergrad, we thought lactate was a negative byproduct, right? Causes problems, changes the acid level, all kinds of issues, leads to fatigue. Well, if I'm working really, really hard and I can't shuttle it out into the bloodstream enough, yes, it does a bit. But generally, the lactate gets moved into the bloodstream and goes off and does something very positive. It's whether or not I've got these, if I'm working so hard here that I'm pouring off hydrogens, I can't pick them all up, then I start to see some fatigue. right? So hydrogen is still the bigger problem. NAD stands for nicotine adenine dinucleotide or something like that. So we don't worry about learning that, we just call it NAD. Because I work with preschool people, I like very short, simple words. Big, long words make me nervous. So we're going to stick with NAD, right?
Thanks, Dr.
How are we? All right, so the next slide really just talks about what I've just told you, okay? And then on Monday, we'll start looking at training adaptations. Okay, so we'll review this idea Monday quickly, and then we'll look at the adaptations to glycolysis if I use that system in my training.